Okay. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone to our honorable lecturer, Mr. Dana Wicaksona, and to all of my beloved friends. Okay, so in this nice opportunity, we are from second group of this class. We want to discuss about disease incidents. But before we speak more and more, so we would like to introduce ourselves. Okay, I am Asthiana Hikmatia with student number 218.
My name is Mulya Gaffari with student number 209.
My name is Santika Immanuela Samosir with student number 221.
My name is Ratu Angkasawati Purboyo with student number 220.
My name is Cherya Azmi with student number 206.
Okay, so in this next occasions, we will present to you guys about disease incidents, but I want to start with the definition about disease incidents. So previously we learned about last week we learned about intensity or disease intensity, and this topic is disease incidence. So there is a difference. So plain disease incidence can be defined as the number of plain units that are visibly diseased, usually relative to the total number assessed. So example determine the proportion of diseased plants per field or the proportion of diseased leaves per plant as representation of incidence. And how do we know about these incidents? So to determine disease incident by assessing or observing the disease status of plant units in one or more sampling units. So a sampling unit can be defined as a portion of the habitable space from which observation that are taken. Next. Okay, so the objectivity or the aim of this topic is usually disease incidents used for measure the number of disease that directly kill plants. So the key concept is that incidence is a inner variable, that is a plant unit is either visibly diseased or not. So in this case, we don't have to know about how severe is disease in that plant. So we just have to know about this plant are attacked by disease or not, just that. So it's more simple. Basically, it's more simple than about the intensity or disease intensity. Okay. Next.
Okay, the next is sampling pattern. There are different sampling procedures that can be used depending on the crop, size of field, and etc. Farmers usually develop their own customized sampling plans once they have experience with a particular crop. Upon entering the field, make a quick visual examination of the field. Look for any atypical areas that might affect the sampling pattern, like the areas with poor stands, of by topographical variation in the field, varietal crop differences, and etc. Sampling can be done in these areas, but any unusual condition that could affect insect numbers should be noted on the sampling sheet. Once we have an idea of the field condition and layout, think about an efficient sampling pattern for the field. For the example, a W or U-shape sampling pattern are more commonly used in square-shaped field. And then for long, narrow field, usually using a zigzag or Z sampling pattern. That pattern are more efficient in that field. The next is for the time. Optimal timing of sampling depends upon the life history and behavior pattern of the pest or beneficial insect and also the crop and environmental condition. Both insect and plants develop quickly under warm conditions and more slowly when it is cold. But in general, it is a good idea to begin sampling as soon as the crop is transplanted into the ground or when plants emerge from the soil if direct seeded. And then the sampling should be done at least once per week and preferably at least twice during warm growing condition. More frequent sampling is usually needed if insect pest number are in the low to moderate range and one increase. It is truly amazing how quickly insect pest population can develop, and once a week sampling may not detect a surge in insect number until it is too late. Okay, next.
And then about disease intensity, in there, disease intensity can be calculated using existing formulas. The things that you need before to know the calculating the formula are the number of disease formulation individual, which is symbolized by letter X, and then you have to understand the number of sampling unit, which is symbolized by the letter N, and then the number of sampling unit can be individual plant if the sample taken an individual plants, but if the sample taken are cluster, then the number of cluster taken is calculated. The formula used to calculate disease intensity is disease incidence is number of disease individuals or X, and then divide number of sampling units or N, and then multiply with 100%. Then this is about an example about the disease intensity. We have to know about total plants in the field. This is 100 plants individual, and then we have to know the sampling unit. There are 100 plant individuals, and then we have to know the number of disease plants, and there are egg plants individual, and then we have to calculate to know about the formula. Egg divide with the 100 and then multiple with 100%, and then we have to get the final result is 8%.
Okay, next to example 2. From 100 plants, the picture is from 100 plants. I took 24 samples of plants that I want to observe. There are 24 samples that in the box, and then from 24 samples, I found 8 plants that are attacked by disease. So the disease intensity is 8 divided by 24 and multiplied by 100%. Then the result is 33%. Next.
So the next one is example using pattern X or cluster sampling pattern X. So just like the title, we make a form shape pattern X. So we, in this example, we make a 5 sampling unit cluster, which is 2 on the top, 1 in the middle, and 2 on the bottom. And every cluster have 4 individuals. So in the first cluster, we found that 4 individuals are diseased, and the second cluster, none of them is diseased, and in the middle, also all of them are diseased, and the third, none of them are diseased, and the last, 3 of them are diseased. So we found that the number of diseased individuals is 11, and the number of sampling units cluster is 5, so 11 divide 5 multiply with 100%, and the result is 20%. Next. Beside cluster sampling pattern X, we also can use pattern Z. We made the form shape pattern Z. So in this example, we have 9 sampling unit cluster, which is every cluster consists of 4 plant individuals. So the number of disease individuals, we found that the number is also 11, and the sampling unit cluster is 9. So 11 divide with 9 multiply with 100%, and the result is 12.2%.
Alright, guys. So that's all for our presentation. Thank you for your nice attention. So if we made any mistakes, we are very sorry for that. Thank you. Assalamualaikum warahmatullahi wabarakatuh.